Πολύ ενδιαφέρουσα παρουσίαση. Εγώ είμαι ο Πέτρος Nihoyannopoulos και πρώτα απ' όλα να ευχαριστήσω τον καθηγητή τον Γιώργο Ευθυμιάδη που για πολλοστή φορά μας εκπλήσει με αυτό το εξαιρετικό συνέδριο και βλέπω ότι κάθε χρόνο μας ετοιμάζει και καινούριες εκπλήξεις. Πολύ συγχαρητήρια και πάλι και σε όλη την ομάδα, γιατί αυτή είναι μια δουλειά ομάδας και όχι ενός ατόμου. I would like to introduce, well, needs no introduction, of course, Professor William McKenna. I know Bill since, well, our very early days at Hammersmith Hospital, 1982, I believe. Professor Vlachopoulos, Professor McKenna is listening to you. And we had all our pleasures and difficulties in the early days, but we worked together and we published together in certain instances. And since then, each one of us went our own way, but still, after all these years, it's so nice to be together in this meeting, and thank you, Yorgos, for creating the circumstances for this gathering. That's one of the personal success of this conference. Bill, we will be looking forward to listen to your clinical application of big genetic data. This is something that is happening, generating new questions and hopefully new solutions for the future. Bill.
Petros, thank you. Thank you for long-term friendship, as you've mentioned. Great title that Yorgos has given me, this clinical application of big genetic data. And I'm sure we all live with two words, big data and AI. And most of us are just simple clinicians. And these imply a whole new future which we need to understand. And I guess our challenge is what data do we want to acquire? What questions do we want to ask of this big data? And to really know that, we need to know where we are now and where we've come from. This is a structure that we all know. And I think if you look back over 50 years, we've used these labels, hypertrophic, dilated, arrhythmogenic, restrictive, as diagnoses. But they're not diagnoses. They're purely phenotypes. And we're beginning to see that under the hypertrophic label, there's 15 or 20 different conditions. And arrhythmogenic, there's at least 10, if not more. So we're beginning to characterize, and ultimately, these are phenotypes in search of a diagnosis, and the diagnosis is genetic. And that's what we are doing slowly, but we're leading towards understanding the familial and genetic basis of these diseases. Big data. I suppose the best, and I'm going to look at hypertrophic, dilated, and arrhythmogenic separately. Certainly from an international standpoint, the UK Biobank, 500,000 people, normal population, is a valuable resource. And people have begun to more detailed phenotyping of this database. But in terms of cardiology, James Moon has led a consortium of looking at cardiac magnetic resonance into this cohort, similarly the genetics. And the big excitement probably in practical terms is genome-wide association, because you need big control populations which are representative of the society, which Biobank is for, at least for the UK, representative. There's other types of information, and I think we see registries. I put up one of them, which is the European Society of Cardiology, and you see the numbers are very small, and at this point, probably limited data allows you to maybe compare the incidence and how disease is managed in France versus Germany versus Greece versus Spain, but very limited in terms of basic understanding. Other types of data, one are these registries, and the SHARE registry. SHARE is sarcomeric hypertrophic cardiomyopathy registry. That's probably the best example, largest example, and best example in hypertrophic cardiomyopathy, and probably in all the cardiomyopathies. It's more than 10 years, it was 2014, but the data that's in that from the genetic and phenotyping is sometimes another decade older. So that clinical genetic phenotype data and outcome data has needed to be curated because different centers, different eras of gene testing. I'll go into that in more detail, but it still has been a valuable resource in terms of understanding disease. Another approach, which was mentioned in a question to the two geneticists yesterday in the first talk, was what about big data? What about industry who actually are doing the gene testing, coming together and putting together big data? And of course, that's like asking two big car companies, Toyota to merge with BMW to put together. There's commercial and other interests. So that is not happening. But one of the biggest is, and I mentioned this one as one of, is Health in Code. And it's a Spanish gene testing company. There's more than 45,000 gene tests in cardiovascular disease, and there's about 12,000 in hypertrophic cardiomyopathy. And this confirms what we actually know from other diseases, that the most of the disease-causing genes are MYBPC3. And you notice the low 23%. That's because this particular registry would have received bloods from clinicians who are not referral centers. They're asking the question, query, is this guy with a funny ECG, is this a hypertrophic cardiomyopathy? So it's a low incidence. If you go to a referral center where the patients are seen in dedicated hypertrophic clinics, you're more like 40%. But that's the background. This is an example of a current example, Nature Genetics 2025, and this is a consortium of Dutch, English, and other groups, and it's a GWAS. You see the numbers, they're good numbers, but then look at the outcome from this. So this is a lot of work, a lot of effort, and we found one new gene, something actin binding protein. So that's a lot of work to find one new gene, and then you see that there's 70 different loci that are associated with HOCM and another 62 that are associated with structural and functional changes. Now, that opens up, what are these other loci? Are these potential modifier loci variants? And of course, that is the question, and that's where we're heading. Thomas Gosius's talk showed a little bit about intermediate effect variants. Other genes, other variants, they may be VUSs, they may be missense mutations that don't cause disease, but they are having some influence, and that's where we're heading, is understanding how these other genetic variants actually influence disease. This is an example from the SHARE registry, and this slide is actually gratis of Tenaya Therapeutics and Natalia Patterson, who works with them. But if you look on the left-hand slide in the survival curve, there's 3 different age groups. So those 3 curves represent 3 different ages. Age diagnosis, age of clinical presentation, 18 to 39, the first curve, 39 or 40 to 59, the second curve, and over 60, the third curve. And you'll note the slope of the curves is similar, but the age of presentation is different. So the message is early age of presentation, adverse features. It's in the obvious questions. Wait a sec. All 13 or 1,600 patients had a truncation in myosin binding protein C. Why the difference in outcomes? Why the difference in presentation and outcomes? That's the key question. And is it modifier genes? What other factors are involved? And those are the questions that we're trying to get at from these registries. Here's an example from the Health in Code database. So these are survival curves for the 4 genes shown: myosin heavy chain, myosin binding protein C, troponin I, and troponin T. And if you look at these 4 survival curves, they're statistically different, but they're not actionable. I mean, you're not going to put an ICD into someone on that red curve simply because they're slightly higher than the blue curve on the bottom. And that's been a dilemma, in that the clinicians who are not enthusiastic about genetic testing have said, gene testing is not actionable. What am I going to do if I know that it's a myosin mutation? Nothing. I'm still going to manage the obstruction or the symptoms or the paroxysmal atrial fibrillation the way I would otherwise. So it's not actionable. This, at one time, that was probably a correct statement 25 years ago. It's not a correct statement now. And if you look here, this just looks at alleles encoding amino acids 92 to 95 in cardiac troponin T. And you see, the bottom curve is that blue curve on the bottom. And the top curve in red is alleles encoding the 271 the 271 alleles encoding the amino acid 271. And they're statistically different, and they're actionable. So, and if you go, and what's happened in terms of outcomes? Well, the light blue is sudden death, ICD discharge. The red is effectively heart failure, transplant, heart failure complication. So these are serious and major adverse cardiac events. And if you look even further and segment by gender or sex, the top line is 271 female. She's fine. You're not going to do anything. If you look at the bottom line, that's a 92 to 95 male. 40% of those will have died suddenly by the age of 50. So clearly you can make an argument that that individual, all by himself from the knowledge of the gene test, you should be considering a defibrillator. So there is genetic information here that's important. And in the past, we've compared apples and oranges. We've compared troponin to myosin, C protein to troponin I. You need to drill down to the specific variant within the gene. And that's what, and this is one example. You can do the same thing with myosin heavy chain, hinge region. The different regions of the gene encode for more severe disease. If we move to arrhythmogenic cardiomyopathy, it's been a different world. And here you see 3 examples, and look carefully at the titles. In each one, these are titles that involve a specific gene and outcomes or phenotypes. And we drill down, and why? Because this community, the arrhythmic community, has actually done genetic testing on their patients. And if you look at the numbers of patients, these are much larger numbers than you would get in any one center. In each instance here, these are collaborative studies between expert centers, and the expert centers have been willing to work together and come up with data, and with important data. And the third topic down, the third slide down, or publication down there, large number of centers, continents, the whole Australian database. It's huge numbers of patients, major collaborations, and that, I think, has that in the short term will be the kind of registry that will be paying dividends. And here the ARVC community, or the arrhythmogenic cardiomyopathy community, is actually ahead of the game versus hypertrophic, which has had at least a 10-year head start. If you look at the last line there, it's gene-based approaches to arrhythmogenic cardiomyopathy in diagnostic evaluation. And this is an initiative that's very important. So we've had in 1996 and in 2010 diagnostic criteria like rheumatic fever, numbers of scores, major, minor, ECG changes, echo changes, a whole bunch of other things, arrhythmia. It's a phenotype. Within that phenotype, there are different genes. Here, and Hugh Calkins deserves major credit. He's the electrophysiologist at Johns Hopkins. He's led an initiative with Fira Duru from Zurich, where they've said, let's make this, the next diagnostic criteria should be genetically driven. They've invited virtually anybody that wanted to join, and by and large, I'd say 95% of relevant people with a lot of experience have joined. There's more than 120 authors in what is going to be an evaluation of 10 different genes, and the paper will go, there's writing groups, there's leaders for the writing groups. If you wanted to join this writing group and that, you could. There have been several meetings discussing the whole thing, and the paper will go to circulation next week. And what will be interesting is the paper is 160 pages long, so it will be interesting to see what circulation does with 160 pages. But nonetheless, there's a fantastic initiative because it's putting the genes at the forefront. And special credit in that If you look back 10, 15, 20 years, arrhythmogenic right ventricular, ARVC, ARVD, ACM, this was a disease of the arrhythmologists, electrophysiologists. And we went through a phase where every patient with a diagnosis had Holter monitoring, electrophysiological study. If you got something going, you ablated it and you put it in ICD. The electrophysiologists and Calkins and Hopkins have led the way. They've actually come back, way back. This is not primarily electrical management, it's prevention. Most of these patients do not need management in that sense. And they've gone back to actually what are the causes, how are the different genes related. So this is a fantastic initiative by Hugh Calkins and many others. One of the interesting things, and this is true regardless of what the gene, what the condition, is the quality of the data in determines what you get out. And the Zurich group have examined their gene testing, and this is a publication in a major journal, and they've looked at 79 patients, and look at the line that says there are 47 variants. So 59% of the variants, when they were reevaluated, they changed the either upgraded or downgraded it as a disease-causing gene. That's putting your dirty laundry out in public, I would say. You're showing that your gene testing has failed 60% of the time or not been correct. And that is a part of the problem, and this has been looked at in a separate way. Cindy James is a genetic counselor at Hopkins, and she's looked at the genes that are reported to cause ACM. There's 26. The 6 are unequivocal. They're all the desmosomal proteins we know, including plus TMEM43 is a gene. It's a membrane gene and found in Newfoundland mainly, and not much elsewhere. PUK is plakoglobin. So she's looked, and they basically said, yes, with 6 we agree with. 2, there's moderate evidence. The 2 are desmin and phospholamban. Those genes definitely cause those 2 conditions, but are they really arrhythmogenic cardiomyopathies? You can discuss that, but basically these are disease-causing genes, whether you put it under this label or that. What's important is that 18 of the so-called disease-causing genes, there's either limited or no evidence, and one in particular, the ryanodine, that is definitely refuted, and yet one of the problems is that the center that reported that still continues to present it as if it was a disease-causing gene, which is confusing to anybody because it's a center of excellence. So there are problems. I show these 2 examples that the curation of the clinical and the genetic information is very important. There's other registries in here. There's all these national registries you can see. China, Canada, the Netherlands. The Netherlands is probably the most developed, and you can see the numbers are small. And on the right-hand side, I put in red what's important. It's actually the familial evaluation is an important component of these registries in ACM. This is DCM, and I think we're probably a decade or more behind. You'll see the same names, UK Biobank, ESC, European Share. Basically, there's very little useful information has come out to date. You see the 2 bottom lines, the pediatric one and Go DCM. These are registries that have been set up, but looking back, it takes a good decade before you begin to get valuable data out of some of these registries. So that's where we're at with that. Here again, there's enthusiasm for GWAS, but then again, if you look, these are the numbers in the DCM, and effectively they found 2 new loci. 2 other loci are confirmed, but it's a pretty small yield, quite frankly, from the amount of work that this would take. So it puts it in context, the amount of work to get something meaningful in terms of our better understanding of the genetics of these conditions. And DCM, our understanding of DCM is well behind. There's other registries, and I've missed the Greek registry, apologies, because it wasn't on ChatGPT. So we need to get it there. What have we learned, and how have we learned it? I would come back and say that what we've learned is exactly what we saw this morning from Nikos and Adelena. We've learned good clinicians studying a family, and that's happened with ARVC. It was Nikos and Adelena. It happened with hypertrophic cardiomyopathy, the myosin mutation, and the hard graft, the hard work. Peter Paré, a chest physician in Montreal in the 1950s, sees a young man with chest pain, breathlessness, and he takes a family history, imagine, and he discovers that at least half the patients in the family have died of either heart failure or suddenly before the age of 45. He creates this pedigree. He finds that the family migrated from northern France to Canada in the 1680s. He tracks the whole family down and he follows it. And he's a chest physician. He's not predominantly a rheumatologist or a cardiologist. He tracks the family. When he retires, he hands basically, this family is from a village I'm from in the Quebec-Vermont border in Canada. He hands the family to Goodwin, who hands it to me. And we ended up studying it. And we discover one weekend that the guys say, you know, there was a family, there was doctors from Boston, they come last weekend, they do the same tests. That was the Zeidmans. So we joined forces with the Zeidmans, and basically the gene was found in the Zeidman lab, the first gene. That kind of work, and that it's similar to what Nikos and Adelena have done. And the families, that's one example. Another example that's very meaningful for me is here's a small family where these 4 individuals die suddenly at the ages shown. And you may say, why do I get on to alleles 92 to 94? Because this family had a variant, had a mutation in amino acids encoding, that amino acids encoded by those alleles. All 4 individuals, the hearts were sent to a pathologist who said, in the Midlands, and he said, oh, the heart's normal. He looked at the coronaries, they were normal, sent the heart to Michael Davis, who's a particularly expert pathologist, and Mike said, this disarray, 40% of the left and right ventricle in the little girl on the left, the 21-year-old, he said, this is extensive myocyte disarray, but there's no hypertrophy. Heart weight is normal, mass is normal. And he writes, is this some form of non-hypertrophic cardiomyopathy? Until we find the genes, we won't know. We find the genes, and that's exactly what it is. And studying the families gives you this kind of information. And that's, for me, one of the things and one of the beauties. Others, just quick examples. This is an atrial fibrillation expert, Patrick Eleanor in Boston. He gets referred patients with atrial fibrillation. He actually does a proper family history. He finds, this is a funny family. They've got HOCM, they've got atrial fibrillation, they've got some other things, and they find a filament C mutation. And in the DCM world, where does the Titan gene come from? Comes from work from Brenda Garul, then working in Germany in Munster with Ludwig Tierfelder, who was then in Berlin. They study a family, that's the first report, and then the Zeidmans pick up. And Titan is a very tricky gene, as you will know, because it's got tons of variants, and it's taken us a long time to understand which ones are actually likely to be disease-causing and which ones not. But again, it's the study of families that has led us to this. Where are we at now? I use hypertrophic as an example, and Thomas Gosius actually showed a recent circulation paper from the people who produced this. It's Soledad Garcia Hernandez, Juan Pablo Ochoa from Health and Code. If you look, we recognize sarcomeric. Yes, these lists are not comprehensive. We look at regulatory and structural genes, FHOD3, TRIM63, and again, there's about 10 of them. They account for between 5 and 10% of disease. We're very aware of the syndromic ones. Then if you look over on the right-hand side, you've got oligo and polygenic risk, and that's minor allele frequencies of less than 1 or greater than 1%. These variants are quite common, and it's clear that they begin to influence disease. So the current work that we're involved in is actually looking at these as determinants of modifier genes which explain the variants in the phenotypes. And that's where we're heading. Understanding the genetics is very important, because once you understand the genetics, you have the potential for therapies, and we see that no more clearly than with the myosin inhibitors. Similarly with gene replacement therapies, this is going to be real. We're in an early stage. We're probably on stage 2 of 10, but this is the rocket experience with Danon in the New England paper, and with PKP2 and ARVC, myosin heavy chain, MYBPC3 in HOCM. This is happening and it's real. Sorry, I lost my finger here. So in summary, I think if you go to the bottom line here, big data, no question. In the future, that's the answer. Where are we now? I think we're beginning to collect those big data sets in registries, in national data banks, and that's happening around the world in different countries. What have we learned so far in the past? There's no question. The families have been the most valuable resource, and thanks to clinicians like Nikos and Adelena, they have been well studied. What's our major limitation? It's been the genetic testing. And a difference, exemplified beautifully, is that in arrhythmogenic cardiomyopathy, the centers recognized early that there's quite variation, and we knew the genes, so we know that desmoplakin is fundamentally different than PKP2 and others. In hypertrophic cardiomyopathy, much more common, the clinicians have been refractory to do the testing. And one major center in the United States, probably the major center, gene tests less than 15% of their patients. Why? There's no money. It's a responsibility. You need genetic counselors. They can't do the work in-house. They have to send it out. So I'm being a little bit sardonic or skeptical, but those are the kind of reasons. Going forward, it's going to be very clear. You cannot see patients without doing the gene testing. So that becomes, and big data become the key evaluating going forward, how we understand disease expression, which is very different. It's not just the myosin, the single mutation, it's the other variants, VUSs or whatever, associated with that variant. So in summary, I think the bottom line at present is that the potential value of big data is very clear, and the importance of genetic testing is very clear. But it's a lot. We're on a big path that will take us a long time, and it's going to take everybody collaborating. And what's very interesting to me is who's been around watching this and involved in the arrhythmogenic cardiomyopathy community. There's collaboration, and all the studies that I showed published, it's 20 centers, 25 centers in hypertrophic cardiomyopathy. It hasn't been collaborative, and that's one of the differences. Thank you.
We thank Professor McKenna for this excellent and stimulating lecture. I have one question, Bill. You clearly demonstrated, and we have been saying that year on year, that it is important to do genetic testing. Why the pharmaceutical industry, while developing new drugs for hypertrophic cardiomyopathy in particular, they focus predominantly on phenotypes, regression of hypertrophy, clinical signs, yes, and the results are good, but they could be better. Is it because they include all sorts of genotypes of genes, and they do not separate each disease, because hypertrophic cardiomyopathy is not one disease? So to me, it makes no sense to produce drugs that focus only on phenotypes, and particularly regression of hypertrophy, and not specifically on genes. I understand. Now we have the opportunity with the big data. Yeah. Put everything on AI, ChatGPT, and can formulate subgroups of patients with specific genes.
Really good question. It's multifactorial. So if you look at hypertrophic and the myosin inhibitors as one example, the companies actually know the variants in those patients. They have that data. It hasn't been published. It's kept in-house. Somebody must know, but it's not easy to get access to that. Why? Because they have been able to sell to the FDA. We're dealing with patients who have obstruction, who have class 2 or 3 symptoms, whose VO2 max is less than some percentage. So the FDA have accepted this, so they're using those endpoints. The truth is, and anybody who's used myosin inhibitors will know, getting rid of the gradient is actually not, that's what the FDA want, and that's how you publish. But that you get efficacy sometimes without getting rid of the gradient, and you sometimes get rid of the gradient and you don't have efficacy in terms of clinical exercise capacity and symptoms. So there's much more to it than that, and the genetics are important. They're very important. And again, it's multifactorial, but part of it is that the HOCM community has not done the gene testing. And it's as simple as that. When Mayo Clinic has done less than 15% of their gene tests, you know you've got a problem. In contrast, in arrhythmogenic, and where the gene therapy is going to drive it is gene therapy. You need to know the mutation. You need to know that it causes haploinsufficiency or poison polypeptide, which determines what approach to gene therapy you take in general. And that is fundamental to actually going forward. And the gene therapy, we're probably in a course of 1 to 10, we're probably at 1.5. But the Danon study in the New England was successful. There's a lot to learn about the immunosuppression if you're using viral vectors. But basically, this is very, if you're putting out money, somewhere over the next 10 years, that's going to work, although I wouldn't necessarily put my money in the next 12 months.
I want to bring in the discussion Professor Franco Cecchi at this point. If you can take the microphone, Franco.
I believe that you gave really a wonderful lecture. The problem is, how do we use genetics in practice, okay? So we started in 1999 with the troponin T, okay? And we now have collected more than 1,000 patients and families. The problem is that we have to reassess, as you mentioned before, about 60% of the variants that were found at that time must be reassessed. And very often, the ones that were pathogenetic are not. The ones that were not pathogenic and benign are now positive. So it's a work in progress. It's a work in progress. It costs money. And I have experiences in Italy that they are terrible. For instance, in Sicily, there are, cardiologists that say any VUS variant of unknown significance is pathogenetic. So you need to implant ICD, you need to do whatever you want. So the cardiological community needs to be really instructed. We need to be to a reassessment of the use of genetics in clinical practice. People get, we saw before, I didn't understand the word, but there was a psychologist before. And, when you do a genetic testing, there is a lot of anxiety. I was surprised when Arthur Wilde, a couple of years ago, when he was retiring, he made a big event. And, I was surprised that in Holland, in the Netherlands, when you have sudden death and then you're going to investigate, the proband, the one that died was, genetic testing positive. They have their phospholambam, for instance, as a cluster. And when they asked the families, 50% of the families refused. So they sent, some geneticists at their home, trying to convince them. They said, no way, we don't want to know. So there is still a lot that we need to do in this setting. Anyway, thank you, because these big data are really informative.
Yeah,
thank you, Franco. Yeah. Are there any other comments? Take the opportunity. Professor McKenna is not here every day, so take the opportunity that he is here. Well, I think, yes, Thomas.
Thank you very much for the excellent talk. Just a quick question about big data. Setting up, let's say, a project. Where would you start from, the genotype or the phenotype?
Sorry, setting up a what?
A project of, a big registry, something that we will start putting that data in. But what would your starting point be? Because I think a key problem is variability, what we call phenotype, variability for how we interpret.
I don't actually see that as an issue in that, what comes through the door to see you as a patient is not an MYBPC3 mutation. It's a patient whose whose brother died suddenly, or who has syncope or chest pain or palpitation. It's a patient with a problem. You characterize the phenotype, and that's why these simple phenotypes, hypertrophic, dilated, and restrictive, have some meaning. It's the predominant feature, and you characterize the phenotype as we do, and then you do the genetics. Now, how you do the genetics, that's the more complex question, and we're not the kind of people to answer that. Is it whole exome? Do you need whole genome? When do you do this? Do you start with candidate panels? And it's probably there's probably different answers for different circumstances, but the phenotype is the starting point. It's not the genotype. It's not a gene that walks through the door. It's a patient with a problem.
Okay, I think if there are no more comments or questions, Yorgos, shall we continue? Thank you, Bill. Thank you very much. That was excellent session.